Okay, so we're going to pick up with a facilitated transport. So let me get my screen up for you folks. Okay, facilitated transport, here we go. So we left off talking about how nonpolar molecules and molecules that and stuff that doesn't have a charge can get through the plasma membrane pretty easily. But what do we do when we have really large molecules, or if we have molecules that have a charge on them? We need to get them in and out of the cells using different methods. And so we're going to talk later about active transport, but still here we're still at the part talking about passive transport. And this particular mechanism is still passive, but it's facilitated. There's some helper proteins in the plasma membrane that allow this to happen. So we talked about how charged particles, whether those are polar molecules or ions, are repelled by the fatty acid tails. That's the hydrophobic portion of the plasma membrane. But there's some proteins that can act as like little tunnels, little pathways, or even like little escorts through the fatty acid tail, the hydrophobic part of the plasma membrane. So let's talk about, remember what integral proteins are. These are the proteins that are like totally stuck in the plasma, the phospholipid bilayer. Part of the protein is sticking on the outside of the cell. And then the whole protein is kind of in the middle. And then there's a part of the protein that's sticking out on the interior of the cell. So we call these transport proteins because their job is to bring stuff in and out of the cell, to transport stuff. And so there's kind of two major flavors. There's channel proteins and then there's carrier proteins. And we'll talk about carrier proteins in a little bit. But right now we're going to focus on the channel proteins. So in this diagram, we can see different mechanisms of passive transport, where we can have simple diffusion, where these small uncharged particles are making it through the plasma membrane. And then here we have a facilitated diffusion, this one using a channel protein. So here, this is an integral protein. So we've got part of it that's sticking out into the external fluid of the cell, so the external cellular matrix. It's a fancy science talk for that. We've got the protein, part of the protein that is stuck inside of the phospholipid bilayer, and then we've got part of the protein that's sticking out. And so it's completely integrated into the plasma membrane, and so that's why we call it an integral. This particular case, there's a little tunnel, a little pathway that our charged particles can make it into the cell's interior by traversing this integral protein. So let's say that these guys are, I don't know, what's our favorite ion? Let's say, I think I used calcium in class, so let's use calcium. So calcium can't get through because it has a positive charge because it's an ion. So, but it can get through this little pathway of this integral protein, channel protein. And then we also have our channel proteins can come in kind of two flavors where they're open, just like we just saw in the last example. So here we have an open integral protein, channel protein again, where these things are coming in and out of the cell, kind of however they want. They're diffusing according to the concentration gradient. And there's no control over that other than it has to go through these proteins. And so here's another example where it's a gated channel protein. And so the cells can actually control whether this gate is open or closed, depending on if it wants that stuff to diffuse or not. Okay, so that's that. So now kind of switching to the other type of integral protein that allows for facilitated diffusion, we call these carrier proteins. Again, we've got this protein that's stuck completely in the phospholipid bilayer. Part of it is on the outside, part of it is in the plasma membrane itself, and a part of it is on the interior of the cell. So I think in classes is where I talked about the calcium. So we could actually even talk about, let's say, call these potassium, or sodium. Let's call these sodium. So these sodium ions can go and stick and live inside of this pocket in the protein. So remember back when we were talking about enzymes, and we talked about how the shape of the enzyme is really important for how it functions. That same concept is true for carrier proteins. Its shape really matters. And so in here, in this example, we have this little pocket where it can like kind of scoop up, or these, you know, these ions are attracted to this site in here, this little pocket. And it's like chomp. And then the way that the amino acids that make up this protein interact with those positively charged or negatively charged ions is going to cause that protein to open up on the other side. It's changing its shape because it's interacting with those different ions. So then it like poops those out into the cell. And then we might see it open up the other way. And so this is how these carrier proteins act, is that they're allowing, they're carrying these ions through the plasma membrane, through those fatty acid chains, and into the aqueous environment on the other side. What this does is that this can limit the rate of diffusion. So like we've all been in the grocery store on, like, you know, a couple days before Christmas or Thanksgiving or something, and there's a lot of people there that are doing their last minute shopping, and it doesn't matter how many more people that are coming in, the amount of people that are leaving is dependent on how fast the cashiers can process the groceries to bring you up, and you pay for it and you get out. So adding more shoppers into the grocery store is not going to increase how many people are leaving. It really is going to depend on how quickly and efficiently the cashiers can do their jobs and how many cashiers there are. By adding more cashiers, we can like process more people more quickly. So that's kind of the same analogy that I want to use here. By increasing the amount of ions is not going to increase the rate of diffusion like we see in like normal passive diffusion, but because they're dependent on these carrier proteins, increasing is not going to necessarily make diffusion happen faster. Okay, so osmosis is a special form of diffusion. So let's talk about, so this is the definition of osmosis given in your textbook. Osmosis is the movement of free water molecules through a semi-permeable membrane according to the water's concentration gradient across the membrane, which is inversely proportional to the solute concentration. Okay, what now? What? So let's break this up into its little, just these little bits, because if we can kind of make these into bite-sized pieces, this crazy definition kind of starts to make more sense. So before when we were talking about osmosis, we talked a lot about solutes and ions and the crap that's dissolved in water. But really here, when we change gears and we talk about osmosis, we're talking about only water molecules. Only water molecules. That's one of the major components that make osmosis osmosis, is that when we're talking about osmosis, we're talking about the diffusion of water molecules. If we're talking about diffusion, we can be talking about any substance that we want to talk about. But osmosis, only water. Okay, and then the next little bit is through a semi-permeable membrane. Well, that's what plasma membranes are, right? They're semi-permeable. There's other types of semi-permeable membranes that aren't plasma membranes, but in our case right now, we're focusing on those plasma membranes. And so we're talking about water entering or leaving a cell when we're talking about osmosis. Okay, the next little phrase is according to the water's concentration gradient across the membrane. So just like with other forms of diffusion, we're seeing water molecules moving from a high concentration to a low concentration. And so what we're do So again, here's our high. So I think these little blue dots are supposed to indicate water. And so we see that on this side of the plasma membrane, the semi-permeable membrane indicated by this dashed line, that there's a high concentration on this side. And so the water molecules are going to move from a high concentration out into a low concentration. And so that's what that is showing, that the movement of water is going from high to low. Okay, and then this last little bit, which is inversely proportional to the solutes concentration. This is just mathy talk for saying that as you increase the concentration of one, the other one goes down. And so these concentrations are moving in opposite directions. If we're increasing the concentration of water, that means we're decreasing the concentration of the solute, or vice versa. If we're increasing the concentration of the solute, which means that we're decreasing the concentration of the water. So this inversely proportional to, that's just like other, like opposite directions. Okay, if one goes up, the other one goes down. It's like an elevator weight, and it's counterweight. Okay, so don't get mixed, don't get frustrated or intimidated by that. Just kind of, because you guys can understand this, when one goes up, the other goes down concept, and that's all that is. Okay, so osmosis is going to occur until it reaches equilibrium, or until the pressure balances. So once there's kind of an equal concentration of water molecules on either side of the plasma membrane, then that means it's there's no more osmosis that's going to occur. Or we might see a little bit more pressure on one side, which is going to cause a differences in concentration of water molecules, which can still be at equilibrium, even though because of one side is under pressure. Okay, so we love new vocab words. So we're going to introduce a couple more. So tonicity is a way to describe how the concentration of solutes and the stuff around the cell can affect the cell's volume by how osmosis is occurring. And so then osmolarity is describing the solution's total solute concentration. So all the potassium, all the sodium, all the I can't think of any other ions right now. Let's say iron ions, all the sugar that's in there, all the salts that are in there, in that dissolved in that solution. We're talking about all the stuff. Okay, and so this is where students can get mixed up. And so make sure that you are spending enough time with the concept of osmolarity to be able to get this straight. So low osmolarity means that there's a lot of water molecules compared to the solute molecules. So the concentration of water is high, and the concentration of solutes is low when we have low osmolarity. Okay, so when we're thinking osmolarity, we're thinking about solutes. And so if we have high osmolarity, we have a low amount of water molecules compared to the solutes that are there. So osmolarity is about solutes. So that means that water is flowing from areas of high osmolarity, where there's a lot of water molecules, to high osmolarity, where there's a low amount of water molecules. So we tend to think about diffusion going from high to low, but osmolarity is talking about the amount of solutes. So and then if we're talking in terms of osmosis, we kind of have to turn that into backwards land. So just remember, osmosis is the concentration of water. So don't get that mixed up. This is a point where it's easily, you can get confused easily. So make sure that you understand this concept down solid. Okie dokie. All right, so tonicity is talking about how comparing the concentration of the extracellular fluid to the concentration inside the cell. So when we talk about these words, hypertonic, isotonic, hypertonic, we're talking about relative to the cell. The point of reference here is always the cytoplasm. So we're going to be cell-centric when we talk about hypo, isotonic, and hypertonic. Okay, so let's explain what these words are. So hypo is means below. So let me go grab my little whiteboard here. So hopefully you're keeping a dictionary or a list of all the Greek and Latin root words that we've been talking about. So that these things are going to, as we go through the course and as you go through more courses in learning biology, you'll be able to pick apart these words by looking at those suffixes and prefixes. So we have hypo, which is just another way to say low. Iso just means samesies. And hyper is high. Okay, so we all know that hyper kid that's like, dude, you need to calm down a little bit. No more marshmallows for you. Okay. So because that kid is high energy, right? All right. So what we're talking about with hypo, we'll go in order of these images. So the hypertonic condition is where there is more solutes on the outside of the cell, which means that the concentration of water is low on the outside of the cell compared to the concentration on the inside of the cell. So then we see the water leaving the cell to diffusing across that semi-permeable membrane. Because we're talking about water, we're talking about osmosis. So that is why we see this exit of water. And so that's when you guys were doing the lab and you looked at the Elodea leaf under the salt water and it looked like kind of like this because the water left the cell in order to try to dilute that salt water. So, we know that molecules don't have sentience. Okay, so this is why we don't drink salt water, because you probably have heard that if you drink salt water, that's going to make you more dehydrated. And this is exactly why you're creating a hypertonic condition inside your cells. The water inside your cells will leave your cells, and then you become more dehydrated. An isotonic condition is where the concentration of the solutes on the inside of the cell are the same as the outside of the cell, and so water movement is at equilibrium. We still have water movement, it's just the water coming in and the water going out are the same. And then our last term is hypotonic, and then this is where the solutes on the outside of the cell are lower in concentration than on the inside of the cell. And so that means that there's going to be water coming into the cell, and then the cells are going to swell. And then if they get too swollen, they might actually be enough to break the plasma membrane and those cells will burst. So I don't know if you've ever cut yourself over the bathroom sink, and as soon as the blood hits the water, it just goes, and then that whole thing is just like, then there's a big mess, right? And that's because the cells are in a hypotonic condition, and they're bringing on more water than they can actually structurally hold. And so we see the cell actually break and lyse. And so here I've already said all these words, so I'll just leave this in the PowerPoint for you guys to be able to look at these. But we have, so this is just for your ease of studying. Okay, so here's the image from the textbook that's showing a hypotonic condition where the water is coming in and actually breaking that red blood cell. Isotonic is that everything's at equilibrium, everybody's happy. And then a hypertonic is when the water is leaving the cell and this cell shrivels up and can no longer function. Okay, and then we can also see this in plants. You guys saw it under the microscopes. And so what this is showing us is that plants lose their turgor pressure. There's not enough water to hold their plasma membranes against their cell walls, and the plants begin to wilt. And some plants can be really dramatic, like this one. Okay, so hopefully that helps clarify some things. I'm going to make another video about the active transport mechanisms here in just a second. So, okay, have a good day. Let me know if you have any questions. And I will see you soon.